I have to say that I've been many times to Hangzhou, that this is the first time I've come to West Lake Shibu, and I'm very impressed by the whole campus. I would like to congratulate all of you for working in such a fantastic environment. So, I've been working for 16 years at the Chinese Academy of Sciences in Guangzhou, and it was in the last couple of years in my interactions with PGI, mainly PGI Shenzhen, that I came in contact in a heavy way with the power of mega data. And this completely blew up my mind. And then it was perhaps the main reason for leaving the academy and Chinese Academy of Sciences. And I moved to PGI Shenzhen in December last year to this new unit, which is Physicist Star. And we work on different things. We would like mainly to create a virtual map that goes from early development until the very end of life, with all the different processes in the middle. We are not to use just sequencing, but also use different types of automated technologies with the goal of learning about these processes, and bringing all this knowledge from the early into the old and the south, not just knowledge. For this, I'm going to give a very simple talk about some of the work that we did in contact with PGI, but before joining PGI. The work that we're doing now, the complexity is significantly much higher, but I think this can give you an idea of where we're heading to. One of the important things that we're doing is we are creating cohorts of humans from different ethnic groups. One of the first cohorts that we have created is with Chulalongkorn in Bangkok. It's the top university in Thailand, and together with our colleagues there, we have created a cohort of 9,000 individuals between 60 to 100 and over 100 years old, and we are following them in a longitudinal manner using omics. Okay. All right. So our team has been originally a stem cell biology team. So we have been interested for quite a number of years in trying to see the different stages of development, how they can be reproduced in vitro. It's quite interesting. Aristotle was the precursor of embryology. For a very long time, everything was forgotten. But then now there is a boom in this field, and this is going to impact significantly in the field of aging. So we just started with a very simple hypothesis, which is to improve the culture conditions for producing cells that mimic the early embryo. And when we applied single-cell RNA sequencing and other type of single-cell technologies, we observed that we could identify cells that mimic the very early stages of human development when the cells, the human genome becomes activated. So we have been working on these 8-cell embryonic cells for the last several years, and we work on 2-dimensional models, specific directed cell differentiation, 3-dimensional models. Embryo is becoming very important clinical research now, and we have created a map of early human embryogenesis. This work is pretty much finished, but I'll talk about this on another occasion. So the power of sequencing, and not just sequencing, because we're going to be moving into, but not just sequencing DNA and RNA, we're going to be moving into sequencing proteins, and this is going to also completely change the way we see things, and together with artificial intelligence. So we are working on the idea of perturbing all these different pathways in early embryo development, perturbing all of them at the same time in different ways, sDRNA libraries, transcription factor libraries, transcription factors that don't exist in that normal shape in vivo, thanks to a DNA and RNA synthesis platform that we can produce all these different types of libraries in very short time. And then using CropSeq, I guess you are familiar, just putting a tag, you can follow, for example, your sDRNA in the cell. We are creating maps of how perturbing these early embryonic cell models, we can produce different types of lineages, and this way learn more about how to create this map of early development. For this, we also use StereoCell, but I will introduce this towards the end of the talk. So we use these early embryonic cells to do plasticity complementation in pigs that we have been doing knockouts for genes related to kidney development, for example, also heart development and other organs. And we were the first group to create a kidney primordial in a pig embryo. This follows the work of Carlos Lemonte and collaborators who demonstrated that when we inject human cells into pig, they can survive. But we could demonstrate that not only they survive, but if you put cells in the right conditions and in a specific molecular niche, it can assemble into structures. This was done in collaboration with our colleagues Lina and Wendy. Okay, so we did the same but this time with monkey cells. It's been a debate about whether monkey cells really achieve the same early embryonic development state as mouse cells and human more recently. And we did plasticity implementation with monkey cells and monkey embryos and homologous embryos. And we were the first team together with our colleagues Lothar and Sunshine and Shanghai demonstrate that we could produce in monkeys with very high contribution of the donor cells. And you can see how the very unfortunately this monkey had to be sacrificed for 10 days because the health was not good enough. This brings us to the point that we still need to improve the culture conditions. But in the case of the brain, using single cell sequencing and other approaches, we could demonstrate that 85% or more of the cells come from the donor cells, and this has a lot of implications. We're working now on how to improve these culture conditions. We've already done. We are improving the chimerism, and we're doing these to produce different animal models. Okay, now moving a little bit into the adult stem cells. One of the first works we did with PGI was this non-human primate atlas. Like you can see here is just 1,100,000 something cells. By that time it seemed a lot, by now this seems like a relatively small number, at least for us. And then while we work with monkey and we work with other mammals, like mouse, I'm going to show a little bit later, is because in many cases getting human tissue, which is our main interest, is complicated. We're trying to solve that issue, but it has taken us a lot of time, and even then it's still, as you can imagine, lots of limitations. So using this non-human primate atlas, we could map LGI5 positive cells in the whole individual, and this is an amplifier of the Wnt signaling pathway. And the Wnt signaling pathway is not only very active in development, it's also very important in controlling the stem cell niche. These have been mapped in the mouse, but in large mammals it had not been done because this is something that is expressed at very low level, and the antibodies are not fantastic for doing this mapping. So it's quite interesting that we could find a population of cells in the distal convoluted tubule, which is a part of the kidney during development that produce the rest of the nephron that maintain expression of LGR5 only in monkey cells, not in human, not in mouse. We think that monkeys probably have some level of kidney repair that is not present in other species, in other large mammals or mice, or obviously in kidney, in fish, even in chicken. There's some level of repair, but in humans there's very little. We're trying to understand what are the mechanisms for controlling these LGR5 expression and sensing of Wnt in adult monkeys to see whether we could bring those discoveries into humans. Anyway, using single cell sequencing, ATAC sequencing, all that type of type and everything is very important, but the problem is that the cells lack a context. So we work with our colleagues in PGI using this technology, Stereosync, in which we can do a spatial mapping of mapping of transcripts. This looks like a painting. Actually, this is a spatially resolved transcriptomic map of a mouse 16.5 embryo. We have been moving from development to different organs, again with the mouse, because it's easier at the beginning at least to do some of the work. We work on liver homeostasis and regeneration. The liver is a very homogeneous organ, but because of the gradients in the lobules between the portal vein and the central vein and the different composition of cell types, the gradients, the transcriptional and proteomic gradients in these lobules are very complicated. So we created this map of liver homeostasis is a whole load of the mouse in homeostasis, of the mouse leader homeostasis. These different layers are areas of transcriptional similarity. We divide it in 9 layers because that's the kind of paradigm that people are studying. The work of Shalem in the Rebbe Institute in Israel, but actually we can divide it into 100 different layers, and this is also how it looks like if you do bidding of these images. So we apply this not just to homeostasis but also to regeneration. We use hepatectomy, also cholestasis. This was worked on in collaboration with our friend Hui in Shanghai, and we demonstrated some specific processes that were known in the transformation of hepatocytes into cells that have the ability to produce hepatocytes, but more interestingly, cholangiocytes, and repair the liver upon damage of the cholangiocyte network. Okay, now I'll move on to the work that we did this year. It was published this year, on skeletal muscle, and then I'll show some unpublished work. I think I'm going to remind you of how important the muscle is in controlling not just movement but metabolic functions in the body. Also, even though it seems a simple tissue at first sight, it's very complicated with different type of, not just the fibers but mononucleated cells in different places, the stem cells of the muscle, the satellite cells. So we decided together with our collaborator, Pura Munoz in Antos Labs. She's a very good friend of mine, to try to create an atlas of sarcopenia. Sarcopenia is an extreme example of loss of muscle function and mass at a very late stage of human life, and is one of the hallmarks of being close to the end of the cycle. So we obtained samples from Chinese and Spanish patients from different ages. Again, this is relatively simple, but by the time we started in terms of number of patients, 20 patients and so on, now we're working on hundreds of patients simultaneously, and then we did single cell ATAC, and we observed obvious things that people knew, like the muscle contains different types of fibers, type 1 and type 2. Type 2 is a quick one, type 1 is the one that gives you strength. And then we observed a well-known increase in type 2 fibers in all the individuals. But the interesting thing is when we start looking at the different populations that appear. So we observe many different things like loss of the neuromuscular junction, all different types of pathways. I'm going to show some activated or non-activated pathways, subphenotypes, close to senescence, although I will not call them senescent cells, stress and so on. But we also observe phenotypes related to abnormal contractility. So we observe a population of cells TNNT2 positive in the old, and TNNT2 is part of the sarcomere of the muscle, but not of the skeletal muscle, is of the cardiac muscle. And then we think that one of the biggest problems that happens in the old muscle, in the type 1 fibers and the type 2 are gone, is that they start acquiring abnormal features, not just of the type 2 fibers also, but also other types of muscles, like the cardiac muscle. And then, as you can imagine, the contractility would not be appropriate, and this creates a vicious circle in which ultimately the whole muscle becomes degenerated, there's atrophy, and then that impacts metabolism, that impacts everything, and then we pass away. So this is a pseudo time of seeing how the features of the type 1 and type 2 fibers are acquired as the muscle ages. In the type 1, the changes are progressive, type 2 very quick. This progressive effect in the type 1 gives us a window of interference. We're working on approaches for targeting specifically these type 1 fibers in primates, for seeing whether we can have approaches that prevent the loss of the sarcomeric identity, skeletal muscle identity, and the atrophy. So we'll observe different types of pathways, very interesting oxidative phosphorylation, and the glycolysis. They seem to be exchanged. So the glycolysis is very active in type 2, but it looks like in the old type 1 fibers, the glycolysis becomes more more activated those type 1 fibers, and in a way, like I was saying, if you lose the type 2 fibers in the type 1, I'm going to try to differentiate somehow and acquire the features that they don't have so that they can do the 2 functions. But obviously, that just creates a chaos, and rather than being, overcome the problem that becomes bigger. We also have the changes in satellite cells with some population of massive satellite cells defined by some markers such as these markers here. And then we think that one of the problems of the muscle in the old is that because of the constant damage that cannot be repaired properly as it is in younger individuals, there's a constant exit of the muscle satellite cells that in the end gets them exhausted, and then as a consequence the repair is inefficient, and then possibly comes with abnormal differentiation features of the myotubes. I won't get into details. We also study the microenvironment, different potential targets for interference, and then we're moving on to other tissues and to other cell types. We have created this peripheral immune cell database. Is a baseline. It contains individuals of all ages, but mostly young individuals. We are doing the same thing with all individuals of different ethnic groups. There's also some papers published, and many are reviewed right now by other groups. But with this baseline, we hope to be putting more data than what we will be collecting from our cohort in terms of mapping how aging happens in a longitudinal manner. Chinese populations, Thai populations, although there's a big Chinese genetic component in Thailand. We are working on modifications of the STL6 technology, not just for doing the spatial transcriptomics in a classical way. I believe that in around 5 years from now, we may see a revolution in sequencing, so that a whole individual will be sequenced in like 1 day, and then this we talk through optimizations of these technologies or other technologies, which we can put millions and millions of cells in one chip and sequence them spatially, rather than going to average 10, 20, 30,000. So, as you can see here, this is it's up to 10 million right now, and theory with 260 million, probably more if we use larger chips. And then one of the things that we're working is not just to optimize these type of technologies, but is how to do the analysis very quickly. I think that once this is going in a regular manner in our team, the problem will be the analysis. And we hope to be creating partners that, together with artificial intelligence, we can put the data in. So one day sequencing a whole individual, all the tissues, single cell level, and especially resolved sequencing with multi-sectioning and so on. Also one day for all the analysis, and then we can spend a whole lot for interpretations and create all the possible hypotheses together with the artificial intelligence that we can imagine. So this is how the cell works. It can give you an idea also of the spatial resolution of the transcripts, and then you can see cell vicinity, and you can get different types of tissues, muscle fibers, all the types of tissues. You smear them on the chip, and then you can look into associations between different cell types. Some cells are individually, some cells are in groups, and then we're working on this to try to define neighborhoods. So show some data now. Our embryonic from our target friend. And don't worry about taking photos, although this is going to be in cells or bioreactor probably within 1 week or 2 days, so you'll be able to read it properly there. So we have genetic and non-human primate atlas brain. So this is in the human. There's a lot of work on human brain aging and neurodegenerative diseases. But the problem is that these samples, these brains are collected normally at a very late stage when things are already in a very advanced stage of degeneration. The brains are collected very late in brain banks. Sometimes the information of how the sample was sectioned is not as good as it should be. So we are doing mega analysis of data in the whole literature, and although very useful, very important, very interesting, we find that the individual variation, as you would expect, is tremendous. So that's why we are continuing working on the multi-brain. I'm just going to be showing the single cell level, but in the spatial. We're moving on to the spatial, and we're expanding the cohort. We're starting with 13 monkeys of different ages, from 4 years old until 30 or more years, and this is important because Some monkeys in captivity, they live normally up to 23 years old, but those that reach 30 or more years is very abnormal, and this way you can see protective mechanisms that control aging. And I'm not showing anything here, but we found some very interesting things. So we can the cell sensors of the aging non-human primate brain. We have mapped also these sensors on the specific slides. This was previously published in Cell by our colleague Chen Hao and Shanghai team. And we have studied cell populations, as you can imagine, with aging primates, and the cells are exposed to stress. They are sensitive. It's not very clear yet which cells die or which cells proliferate more microglia. I think these are very open questions. But there's some consensus that SSD possibly keep neurons in the cortex and increase the observed sensitivity. We also observe a different type of subtype, which is the deep layer neurons, excitatory neurons, also increase relatively early in life in these monkeys. We don't know whether this is an effect of aging or is an effect of brain maturation or a combination of the two things. We haven't observed the same mechanism in humans when we analyze data, but the variation is so tremendous that we cannot really conclude. And when we have been looking at immunostimulating fish data of the alien brain atlas, actually the staining suggests that the same happens in humans, but we cannot conclude. Anyway, that's not the goal of our story. In the movie we have demonstrated this. We have a larger number of individuals now that we have validated findings. We have demonstrated, we profile regions of different parts of the brain, from the encephalon to the rhombencephalon, and then we have been looking into the subtypes that change the most. The pons and medulla, which contains different nuclei opposed to the cerebellum, is a very important part of the rhombencephalon that connects different parts of the brain. Many fibers go through there and is essential for many different functions. Contains a lot of white matter. The white matter is a very important target of aging, and we have seen that it's one of the parts of the brain that is more affected. Anyway, the genes that are affected in different regions are in general subtype specific. Normally, down regulation is more than upregulation, except at the very late stage in these individuals, like I was saying, 30 years old or more, that we see that the number of upregulated genes in those individuals is more than the downregulated. Many of those genes have protective function or are related to neurodegeneration when they go down, so we have found some interesting mechanisms. In terms of the genes that are shared, different areas don't share too many genes, but the prefrontal cortex is the one that we can neurons share more mechanisms. Many of these genes are related to synaptogenesis and structure and function, and we have observed that some of these genes are unique of the prefrontal cortex. Synapsis is affected in other areas, but normally what implies all types of genes. So we have a very systematic demonstration of the pons and medulla. This is a very important target of aging. Two different mechanisms, microglia activation, but also a normal mirroring repair. And then we have been also studying the regional cellular crosstalk, different mechanisms. The beta is one of the mechanisms that is affected. PPGF, which is regulating the OPC to differentiation. And we have also been looking at the chromatin accessibility, identify different areas that are close with aging, like you can see here in this CAM2 kinase related gene that is very important for controlling synaptic function. The different peaks are differentially regulated in neurons for different parts of the prefrontal cortex. We have studied the same thing in the old monkeys, and we have found that there's a series of peaks that rather than going down in the old monkeys, they go up, and some of these things are related to autophagy and mitochondrial function that we heard in the previous talk. So even for example, that has been linked to long life in Drosophila is one of the things that increases in all the individuals, and then we have found the different transcription factor networks and so on. I think I just want to give you an overall simple idea of what we have been doing. So we also observe astrocytes phenotypes, but we see that astrocytes 1 and 2 in the prefrontal cortex, they are relatively different. It's very interesting because astrocyte 2 is related to aging in mouse, so it seems that in the prefrontal cortex at least is the same thing. So I would like to thank you, and if there's time, I'll take any questions.